cleavage of the zygote, blastocyst formation and implantation. About 24 hours after fertilization, a series of rapid mitotic cell division called cleavage take place in the zygote. Cleavage of zygote begins in the orbita, moves forward towards the uterus by ciliary and peristaltic movements. Cleavage continues, forming a small ball of cells, morula. By the time the embryo reaches the uterus, about 3 to 4 days after fertilization. Morula floats within the uterine cavity, gets nutrition by endometrial secretions. About 5 days after fertilization, a large fluid-filled cavity is formed, surrounding the ball of cells. With the formation of the cavity, this developing stage is referred to as blastocyst. Further arrangement of these cells in the blastocyst results in two distinct structures: inner cell mass and the trophoblast. Inner cell mass is located internally, eventually develops into the embryo. Membranes enclosing the embryo are called amniotic sac. Trophoblast, which is the outer layer of the cell, will ultimately develops into the fetal portion of the placenta. Around 7 days after fertilization, blastocyst attached to the endometrium of the mother's uterus. This is called implantation. As the blastocyst implants, inner cell mass orients towards the endometrium. Trophoblast grows outward, invades the endometrium. This is initiated by the enzyme secreted by the trophoblast to break down the uterine lining. Then, trophoblast extend the finger-like projection into the endometrium. Trophoblast begins to secrete human chronic gonadotropin HCG hormone, which has the action similar to LH. Hormone HCG rescues the corpus luteum from degeneration, sustains its secretion of progesterone and estrogen, which maintain the uterine lining, preventing the menstruation. After implantation, 3 germ layers are formed in the developing embryo at the end of the gastrulation stage. Extra embryonic membranes begin to appear, which surround the embryo. Placenta is formed by the cells of trophoblast and the adjacent endometrial tissues.